Alright guys, so day two of the conference here. Main thing for me, I presented my poster today, and so I'm just going to walk you guys through my poster. And so, if you've never been to a poster session, basically a poster session at a conference, everyone just has their posters showing all of their data, like this is my poster. So literally at SFN, it's the biggest poster session I've ever seen by a large margin. There were hundreds upon hundreds of posters just as far as you could see, and so people just kind of wander around and check each other's posters out and have discussions about what sort of research that they're doing, and so it's a pretty good time. It's pretty cool, pretty exhausting. 4 hours of just talking about my research and talking about what I had on my poster. And so rather than trying to show you my poster and spread it all out, there's no way to ever see it. So I'm just going to put it up on screen for you guys here. Let's see. So there we go. So my poster, the title of my research was Hippocampal damage impairs recognition memory broadly, affecting both parameters in two prominent models of recognition memory. So confusing title, but if you guys remember from yesterday, the hippocampus is this part of the brain that's kind of nestled down in the middle of the medial temporal lobes. And so it's been a question for researchers if the hippocampus is important for recognition memory broadly or for a specific part of recognition memory. And so we're going to go ahead and start in on checking out what I have on my poster here. So for some background here, recognition memory is dependent on two processes, recollection and familiarity. So recognition, first of all, is just the ability to see someone or something and recognize that you've seen that thing before. And so recollection is the process that allows you to recall specific events and details about a past episode, whereas familiarity just gives you a simple old-new memory, lacking any sort of episodic detail. So an example of familiarity would be you see somebody and you have that experience of knowing that you've seen them before, but you just can't place them. And so it's debated whether the hippocampus is important for both of these processes or for recollection alone, and a lot of people have theorized that the hippocampus is just important for recollection. So, two models are commonly fit to recognition memory data in order to characterize the memory signal. And these models are the dual process signal detection model and the unequal variance signal detection model, or DPSD and UVSD. Now, I realize most of you are probably not modelers and probably are not, super familiar with these models. But the important thing here is that both of these models yield two parameters of interest. So it's been hotly debated which one of these models is better. But in the current research, we were just trying to set aside the relative merits of the models and try and talk about whether these models seem to suggest that the hippocampus is important for a general role, serves a general role in recognition memory, or whether it serves a specific role in recognition memory. And so, like I just said, both of these models produce two parameters of interest. The dual process signal detection model produces direct estimates of recollection and familiarity. And the UVSD model produces an estimate of the ratio of the standard deviation of the target distribution to the standard deviation of the foil distribution, or sigma target over sigma foil, and D, just the distance between those two distributions. And so it's not really important to try and understand exactly what those models are trying to say. What's really important is just to keep in mind that both models produce two parameters of interest. So we're going to try and investigate how do the two parameters of each model, how are they affected by hippocampal damage. So participants in this study, we had 5 memory impaired patients with bilateral lesions to the hippocampus. And so this figure that we're looking at right now is just showing you the percent of volume that the patients had compared to healthy controls. And you can see that the hippocampus has a substantial reduction in volume compared to the parahippocampal gyrus. The area of the brain just directly adjacent to the hippocampus was relatively preserved. And so what this tells us is that the hippocampus, and these are measured with magnetic resonance imaging. That's how we managed to measure how big the hippocampus is. And so what this tells you is that since the hippocampus is fairly atrophied, and the parahippocampal gyrus, which is what's right next to the hippocampus, is not atrophied, we can kind of look at that and see that it's a fairly selective lesion to the hippocampus. So so this is going to be good, because now we can see whatever these patients cannot do, we can kind of infer that that's something that the hippocampus was responsible for. So in experiment 1, get that on there. There we go. In experiment 1, we tested the patients 6 times, and the tests were always the same. They would have 50 words that they had studied previously, and 50 words would be new. And so they would just be trying to differentiate which words were the old words versus which words were the new words. And we tested them 6 times in order to get robust data so that we could do individual level analysis. And we tested our controls only 3 times, mostly because we had more controls, and so there was less need to test them multiple times. And so we tested all of our participants multiple times. For the test, participants were asked to not only decide if an item that they were looking at, a word that they were looking at, was an old word or a new word. They were asked to do this along a confidence scale, where 1 indicated that they were sure a word was new, and 6 indicated that they were sure the word was old. And the delay between studying and testing was 3 to 5 minutes. So they'd see a list of words, 50 words, wait 3 to 5 minutes, and then see a list of 100 words. 50 of them would be the words from the study list, and then they'd be differentiating which ones are old and which ones are new. And they did this several times. And so now, on to the results. So if you remember, there were 2 models, and each model yielded 2 parameters of interest. So first we're going to look just over on the left side of this figure, just looking at the discriminability scores from both the patients and the controls. So controls here are shown in red and patients are shown in blue. And we can see that the discriminability scores from both models, D from the UVSD model, unequal variance signal detection model, and familiarity from the DPSD model, both of these were reduced. And in addition, we saw that the variance ratio from the UVSD model and the recollection estimate from the DPSD model, both of these parameters were reduced. So what we can see here is that it doesn't matter which model we use. Both models are showing us that there's a broad deficit to recognition memory. So basically, these patients who have hippocampal damage are showing a broad deficit. They don't seem to have anything selective about the way in which they the way in which their recognition performance manifests on this test. So we wanted, though, to see is this performance similar to that of healthy controls when the healthy controls have weak memory, or is there still something kind of weird going on here that maybe we're not seeing in this particular experiment? And so in experiment 2, we had five test lists that were constructed just the same way as in experiment 1. Again, there's going to be 50 study words and then 50 foils or 50 new items on the test list. And our participants here were healthy volunteers who were tested at 5 different delay intervals: 5 minutes, 1 hour, 1 day, 7 days, and 30 days. And so the purpose behind these variable delay intervals was to try and weaken memory across these longer delays, so we could look at, well, how does memory function in these normal participants when their memory gets weaker? And again, they were asked to rate their memory on confidence scales, and we fit the 2 models to the data, and we found that just as we had seen in experiment 1, all 4 parameters in both models go down as memory gets weaker, which is not hugely surprising. And so that seemed to recapitulate the things that we'd seen in experiment 1. But and now this is kind of a technicality. In experiment 2, just because it would have been prohibitively difficult to collect a lot of data, especially at the 30-day delay. Imagine testing people multiple times at 30 days. That would mean, for each individual test, you'd need a full 30-day retention interval, and it would just end up taking prohibitively long. And so we didn't have enough data for individual analysis in experiment 2, which meant that we couldn't directly compare our results from experiment 2 to the results that we had from experiment 1. So that's what we really wanted to be able to do. And so in experiment 3, we decided that about a 7-day delay would probably be about what we needed to match the performance of the controls to the performance of the patients from experiment 1. So what we did was we tested healthy controls at a 7-day delay, and we did this 3 times. So 3 times they would come in, they would study, we'd wait a week and test them, and then we repeated that process 2 more times for a total of 3 tests at a 7-day delay. Again, they're just trying to memorize words. And so now what we see, so we've got the data from experiment 1 reprinted here. So in red we see controls who were tested at a 3 to 5 minute delay. In blue, we see the patients who were also tested at a 3 to 5 minute delay. And now we've added in orange the controls who were tested at a 7-day delay. And what we see is that controls tested at a 7-day delay, for the most part, match the patients who are tested at a 3 to 5 minute delay. And the fact that these controls are able to match these patients further underscores what we saw in experiment 1, and further suggests that basically what we're seeing with these patients who have hippocampal damage, let me just scroll down to the summary and conclusions here. What we're seeing here is that these patients with bilateral damage to the hippocampus exhibited a broad deficit in recognition memory in experiment 1, affecting the parameter estimates of both models. And then the parameter estimates of both models were also reduced in healthy volunteers as memory became weaker during normal forgetting. We saw that in experiment 2. And then, according to both models, the performance of patients tested after a short delay, experiment 1, was similar to the performance of healthy volunteers tested 1 week after learning. So basically, this suggests to us that that the type of impairment that you see after hippocampal damage is really just a quantitative difference, not a qualitative difference. And so it's a broad deficit that you see in these cases of hippocampal amnesia. And so that's interesting because that implies to us that the hippocampus does more than just recollection. The hippocampus is actually involved in both recollection and familiarity, and kind of serves this really general role in recognition memory, in recognition memory generally. And so that's the research that I've been working on recently. And thanks for checking it out. All right, bye guys.